Hi, welcome to session 5 of PhysiCell workshop and hackathon. In this session, I will talk about custom variables, parameters, microenvironment, cell definition searches, and dictionaries, and everything in C++. So far you have done mostly the work in Model Builder, and with Model Builder, you can pretty much get 80% of what you need to run. But the 20% very specific things that you need to do that are achieved using custom functions, where you can have your biological hypothesis and uh, everything very related to every cell's behavior. So this is just an introduction where in C++ 5 you will go and access these things. In the next section, session 6 tomorrow morning, you will get the full detail of a complete project with custom functions and custom variables. So I will talk about working with C++ defining custom variables, querying cell definitions, querying for microenvironment, boundary conditions, and dictionaries for signals and behaviors. So first thing is, why do we need a C++ thing to add when we can do most of the things in Model Builder? So the simple answer is custom functions, because custom functions need implementation in C++, and in custom functions we access everything in your C++ files. The other thing is, sometimes we have added some custom variables in your model builder, but later you realize, oh, I didn't added one more variable, an alpha and beta, something to some kind of parameter that needs for the function, and you can add it later in the C++ file once your XML file is fully developed. So where will we add your C++ code? So these are the files that you would add it: custom.cpp and custom.h. PhysiCell settings.xml, that is the one that is generated by Model Builder. You mostly when you are editing files in your PhysiCell project, you use these three files. And in out of these three files, the first two, these are the C++ files, custom.cpp and custom.h. They would be found in your custom modules custom modules directory. In the PhysiCell main directory, you'll go to custom modules, and there they would be once you have loaded your project to it. So if once you load the template 2D, template 3D, or any of the sample projects in custom modules, you will see custom.cpp and custom.h. Okay, so first thing we need to add is custom variables. Custom variables are variables that are attached with phenotype, but they are not predefined in the PhysiCell. So for example, in PhysiCell, you have pre-attached things like volume or motility variables, where it is motility direction, what is the speed. These things, they are already there with each phenotype. But then we need to add something more about that particular uh, cell types. Then we will add it in custom variables. For example, we can have an energy variable for each cell. So we can add an energy variable with some dimensions there. So two ways we can add it. One is quick builder, that you can see, you can just go click on it and then add it. And the second that I will talk about is to add it in C++ file. So how will we add it and where would we add it? We will add it in custom modules custom.cpp. That's where we will add the function. And for example, we want to add a variable named energy to the to your cell defaults. So we'll say cell defaults dot custom data dot add variable. So this is custom data. We will add, you can add as many variables as you want. And here I added energy. Dimensionless, these are the dimensions for right now I added dimensionless. You can have some dimensions for it, and 0.5 is the default value for that. So this is how it will be initialized. The other thing is, you can have your custom variable in the XML file. At the end of it, there are user parameters. So you can have that in user parameters and read it from there. So you can add a variable, name is energy, dimensionless, but the initial energy is defined in your XML file. This way you can easily change your initial value without changing it in the C++ code. Once you have written this line of code, it will go into the parameters section in XML file and see this variable name, cell default initial energy, and that will be the initial value of for this variable. Then I have added another variable, you can say alpha. There's no dimensions, and again it can be in your parameters, or you can start with some value like 0.5 or 0.1, whatever works for your hypothesis. Okay. So the second thing is, how will we access these custom variables? We will access these custom variables by in custom.cpp. Everything is in custom.cpp. You can use you can find the index and then you can access. For example, you can say ne is the index of this variable. So you would say PCell, its PCell is the phenotype. So pointed to the phenotype, custom data dot find variable index energy, and that's the index. And on that index, you can find that variable in the in your C++ file, and then you can, in your custom functions, you can do whatever more processing you want to do with that variable. Next thing is cell definitions. So cell definition is you can access them by index. So here, oh, I can see an arrow just like this is a variable, and in that case, I shouldn't have 0 here. I should have more than 0 because it's returning a vector. But we can access the cell definitions by index, or we can access the cell definitions by name. So cell definition by index, we can get all the cell definitions that are 0, 1, 2, 3, 4, and then we can also access them by human readable name. So if you want to file the cell definition of blood vessel, find underscore cell underscore definition blood vessel, and it can give you that cell definition. Okay, so this is about the custom data in cell definition, cell phenotypes, and how do we add data to it. Then comes to the microenvironment. We have discussed the microenvironment in session 3 briefly, where we told you how we add the boundary conditions, and there are 2 boundary condition Dirichlet and 1 Newman. But sometimes inside the simulation, each cell wants to access its microenvironment. So how can it access that? First, it can get the voxel index. So voxel index, this function get current voxel index will tell you your cell's current position. So PhysiCell's background microenvironment is divided into voxels, and we want to access in which voxel our cell exists. And so this is the function to get that particular voxel. And then we want to see that in that particular voxel, what is the chemical substrates concentrations there or gradients there. So for that, we have to get the nearest density vector. So nearest density vector will tell you what is the density vector of all substrates in the environment. But if you want one particular, for example, there's a chemical resource in your microenvironment. So first you find the density index for that. So microenvironment.find density index for resource, and it will give you at which index your this particular resource or this particular substrate is. And if this substrate is not present in your simulation, then it will return -1, which is like an error that this is not present there. So then you can get nearest density vector, and given the variable index, you can access what is the concentration of that particular substrate at that position. So in short, we have a cell. We have to find in which voxel this cell is, and then in that voxel, what is the concentration of each substrate. So that's how we will find it. And also, if we want only one substrate and we want that particular value of that particular substrate on that particular voxel, that's how we get it. We will get the nearest density vector, and we will get the index 2, whichever is on the index 2, it will return us that variable. Okay. So each cell can access the gradients of substrates in its voxel. We can get nearest gradient, and 2 is the gradient of substrate number 2. So gradient substrate number 2 means that there are at least 3 gradients, 0, 1, 2. So there are at least 3 substrates present in your microenvironment. If there's no substrate present and 2, maybe there are total only 2 substrates, then 0 and 1, then this will give you an error. Also, we can access the mesh, which is all the voxels in your microenvironment, and you can access the mesh microenvironment.mesh or microenvironment .mesh.voxels. This will return all the voxels in your simulation, and you can iterate through that mesh, and you can, if you want to add something, you can add to each voxel, or you want to remove something. So this gives you the more refined control on your microenvironment, other than diffusion, that if you want to change go to some particular voxel and change that, or want to see what is there in that voxel, you can access that. Okay, then there are Dirichlet nodes. So we know that Dirichlet conditions can be directly added, and that we can change them for each side in your domain, and that are by default available in XML. But sometimes you want to add a Dirichlet node at some particular position. So it can be that at one point in your Dirichlet in your environment, there's one point where there's a source or there's a sink where there's always some particular value of each substrate at that point. So for that, we can add the Dirichlet node somewhere where n is the voxel number. So you will give the voxel, and at that voxel we will give you a vector where vector is of the size n, and n is the number of substrates in that environment. So if you have 3 substrates, the vector would be of size 3, and it would be a double vector, and it will initialize the values at that point with these vectors where substrate 0, substrate 1, and substrate 2. And so that particular point, or that particular voxel, would act as a Dirichlet node. And this is useful if you want to have some particular conditions somewhere in the middle of the microenvironment instead of the boundaries, because it's easier to do the boundaries directly by XML. But if you want to do something in the inside, then you can use these Dirichlet nodes. You can make few of these Dirichlet nodes at different points. They can act as source or sink for one particular substrate. Okay. Next is the newly added signals and behavior dictionaries. This is a very interesting new thing that we have added to our PhysiCell, and this helps us control the behavior of the cells. So if we think about it, the cell phenotype gets some environment signals, and into that environment signals, it behaves in a certain way. So those signals, so we have added 2 standard dictionaries. One is a dictionary of signals. They will act as input to intracellular models, to your rule-based models, whatever the rules are. Whether they are chemical rules, they are contact-based rules, these are the signals. So this is the dictionary of signals. And then there's a dictionary of behaviors. So these behaviors are, and once that signal is detected, what a cell do with that signal. So that cell phenotype will react to certain behavior. For example, it is chemotacting towards one chemical, or it is chemotacting away from that chemical. It detects certain signal and it starts proliferating. It stops proliferating. It starts differentiation. So different kind of your hypotheses that are there in the biological system that you can add to this signal and behavior dictionaries. These signals and behavior dictionaries are automatically constructed in the beginning of new cell simulation, and then we update them according to whatever combinations of signal and substrates and cell types are present in that particular simulation. So what are the common signals? Common signals are extracellular, intracellular substrate concentrations, for example, like concentration of oxygen. So if there's more oxygen, they are proliferating. If there's less oxygen, they're going into hypoxia and they're doing something else. Substrate gradients, chemotacting towards and away from a gradient maybe. What do we do when we come in contact with a dead cell? So it's like we can have a macrophage, or who can come into contact with a dead cell, or it can remove it, or if you can destroy it, or if coming in contact with dead cell, you can absorb its contents. So contact with other cells. So if you are immune cell and you contact with a cell type, if with an infected cell, what is the reaction that will go on between them? So these are the signals. Then if there is damage, what is the pressure? So if there are a lot of cells, cells are fully packed and there's a lot of pressure on each cell, what should be the behavior of the cell in that particular thing. So these are some common signals. If you want to see a dictionary, you can say display signal dictionary, this function, to quickly see what signals are available right now. And then these are the important functions that we use to access these signals. You can find the signals by index. You can find them by the name of the signals, and we can display the name of signal with the given index. We can get a vector of all signals, get a vector of all cell contact associated signals. So these are different variables how we can access these signals, and we can get a single signal cell for each cell type. So these are different things that you can do, and for most of these, we in the second half especially, there's like cell static B cell. This is pointer to a phenotype, so we can give a cell type and it will return what is the signals associated with it and what are we doing with it. So these are the signals, but the more interesting part is once that signal is detected, what we do with it. So then we have the behaviors dictionary, as I said. So it's a behavior dictionary. Again, it acts as the output to intracellular rule-based model, automatically constructed in the beginning, just like signals. And these are the major classes of behaviors. By detecting certain signals, you can start secretion, we can stop secretion, we can we can change the uptake rate, we start uptaking more, we slow down the uptake. Then cycle progression, cycle progression can be seen as like cells start proliferating and cells stop proliferating. Death rates can be changed, motility parameters can be changed, motility speed can change in response to certain signal, and chemotaxis, cell adhesion, cell BM adhesion, phagocytosis, attack rates, fusion. So different type of rates that are there for phenotype, that can be the behaviors that can be changed by detecting a certain signal. If you want to see the behavior dictionary, you can get that by using this function, display behavior dictionary, and it will give you those signals. Okay. So what are the functions to get those behaviors? You can find behavior by index. You can find behave. You can find behavior index by giving the name of the behavior, or we can find the behavior indices by giving a vector of behavior names. We can get the name of the behavior by giving the index, and simply for empty vector for full set of behaviors. Then we can set behaviors. So for a certain phenotype, we can set different behaviors at different signal signal indexes, and then we write the selected set of behaviors, and then we can set selected behaviors and set single. So we can set one single behavior at a time, or we can have a full vector of behaviors that we can add at the same time. So these functions will help us to attach behaviors to signals, and that make it very easy for us to access them into the C++ files, and you can just write a very complex code in few lines of code. Here is one example that I chose for this lecture, but you can go and see into the interaction sample project. This is not sample project. In the interaction sample project, it is in the sample projects directory. You can see there are certain cells, and they are high rate of proliferation rate unless it is contact with differentiated cells. So this is the function that this is the behavior that we want to model. So we want to model that when we come in that particular cell, which is a stem cell in this case, when it comes with contact with a differentiated cell, it's reduced proliferation. And so what we are saying is high rate of proliferation unless it is in contact with differentiated cell. So we will say we have a half max for cycling, and we will get that for that. So PCD is that particular cell for which we are doing this. For this cell, the base value is for that base value, which we are saying phenotype is cycle data dot exit rate 0. So which is like what is the base value for this behavior, what is the maximum values, and then signal is how many number of differentiated cells are there. So we will add some code we will have some codes before this to find the number of differentiated cells that are present in its neighborhood. Then it's half max, and then we have a hill function to respond to that half max. So what is the signal? Signal is number of differentiated cells, and this is the behavior. And the behavior is to change the exit rate from the cycle. So we are reducing the uh, cycle rate, and we will using that by adding the base value. In the base value, we will add the maximum value minus base value into hill, which is the normal your hill function. And using this function, we have attached a behavior to a signal. So the signal is number of differentiated cells, and we have the behavior is to to reduce proliferation rate, and that's how we can get that. So that was a basic introduction of not all, some of the things that you will do with C++. It will be more clear in the next section when you would use a concrete example to do that. But the important things to notice is that in any customized cell functions, you can access the microenvironment yourself. So you can use the voxels and meshes and substrate indexes to check the gradient of each substrate, or what is the value of that substrate in particular voxel. The second thing is, for best practices, please don't use the hardcode index substrate, because index substrate can change. If somebody deletes a substrate or adds a substrate, your substrate indexes can change, and that your code will break. So every time you get to the substrate, access it by index, access the index, store it in a static variable, and then use it. And the third thing is that behaviors and dictionaries, they are important, and then use them for your they are good use for attaching signals to behaviors, and they are good for when backtracking, when you say what signal is affecting which behavior. And these are the funding acknowledgements as usual, and thank you.